workshop. Um, Yesterday we had a day one, and looking at so many fantastic talk. Today we'll continue with two more sessions to then close the symposium style of the workshop. And then Wednesday, Thursday, and Friday will be practical activities happening across multiple sites in West Africa, in Nigeria, in Ghana, in Togo, and also in multiple sites within Nigeria. This today we are going to be focusing on two sessions: knowledge exchange and industry, and we're also going to be looking into crop and livestock improvement and health. Dr. Ubong Peters will be opening the session today as the keynote speaker, and he will be talking about decoding cancer, and of course, looking into molecular pathways and the future of drug development in the genomics era. Dr. Peters is the lead clinical scientist oncology at Genentech and is a member of the Roche Group. Dr. Peters, we are really happy to have you here and to speak to so many African scientists. This is the African BioGenome Project that aims to sequence at least about 100,000 endemic African species, and this is happening locally on the African continent. Over to you, please speak to us about your work and also the future of drug development in this era.
Yeah, thank you very much for that kind introduction, and good morning, everyone. I'm really grateful for the opportunity to join you at the African BioGenome Project, West Africa's regional workshop. Today's keynote will explore how genomic sequencing is transforming our understanding of cancer and revolutionizing how we discover and develop and deliver medicines. And this has ushered in an era of precision medicine where we aim to give the right drug to the right patient at the right time. And you might even add the right dose. So by the end of this session, I hope you'll walk away with a clearer understanding of the genetic and molecular origins of cancer, and you'll gain some insights into how genomic medicine is changing cancer research and treatment. And we'll also look at how drug discovery and development is evolving in this genomic era. And we'll get a sense of the opportunities and challenges for applying genomics in Africa. All right, here are my disclosures. Before we get technical, I'd like to briefly share this slide, just talking about my journey from my early education in Nigeria to my current role at Genentech here in South San Francisco, California. And this is really geared towards the students and the trainees in the room. I hope my path reminds students that success is possible. So with hard work and perseverance and focus, doors will open, and sometimes the world will meet you halfway. So just keep working hard, and I hope this inspires you to continue that journey. All right, so let's look at the pharmacy of the pre-industrial era before we dive deep into the current era, this genomic era that we are in. So let's take a step back into history. And before science-based regulation, the drug market was really a wild frontier. So you had products like the Hamlin's Wizard Oil and Clark Stanley's Snake Oil Liniment. They were advertised as miracle cures, and there was absolutely no scientific backing to justify those claims. And so some of these medicines contain dangerous substances like cocaine, morphine, atropine, even alcohol. And they were marketed for children, as you can see this child here in this advertisement. And so these absurd claims and drug labels were outrageous promises. You know, like for example, you had here where it said, there is no sore it will not heal. And they're talking about how it is magical in its effects. These were not treatments. These were marketing gimmicks. But that led to a turning point with the birth of the FDA back in 1906, when President Theodore Roosevelt signed the Pure Food and Drugs Act, creating what we now know as the US Food and Drug Administration, the FDA, which would be equivalent in Nigeria to NAFDAC. And that really marked the beginning of science-based drug regulation to ensure that patients who were taking these medicines were safe and the drugs were efficacious and were effective. So today, drug discovery follows a very rigorous process. It begins with target identification. So what molecular mechanisms are we trying to interrupt or modulate? And then we move into hit finding, then to hit to lead, and then lead optimization, where chemists and biologists refine the compound structure and properties. Promising candidates then move into the preclinical testing. This is often done in cell lines and animal models. Um, and so if the data supports safety or potential safety and effectiveness, we move into human clinical trials. And human clinical trials have three phases. There's phase 1, phase 2, and phase 3 clinical trials. And if the trials meet the endpoint that we define, it's a predefined endpoint at the onset of the clinical trial. If those trials meet those endpoints and the experiments and trials were done to FDA standards, then we can submit for regulatory approval. So at each of these stages, we're asking ourselves the questions, is this drug safe? Does it work? And should we move forward? So that's in a nutshell how the drug discovery and development framework looks like today. It really takes a multidisciplinary effort, and modern drug discovery and drug development is highly collaborative. And it takes a team of epidemiologists, molecular biologists, chemists, toxicologists, data scientists, project managers, clinical researchers, and many more coming together to work on a specific drug. And one thing I would like to see is, because I know Africa is rich in talent, we have all of this expertise in the continent. I would one day also want to see us channel this expertise into creating a fully African-owned drug discovery company by Africans, for Africans. And that's something that we can definitely do. So let's transition to cancer and narrow our focus to breast cancer. And I'd like to briefly talk about what cancer is, how it starts at the molecular level, how we classify and treat it, and where gaps still remain. So cancer actually starts when DNA damage in a healthy cell leads to mutations, and those mutations hijack normal cellular processes, causing uncontrolled growth and tumor formation. The key hallmarks of cancer include uncontrolled cell division, angiogenesis, which is the formation of new blood vessels to feed those tumors. You have resistance to cell death or apoptosis, and the evasion of growth suppressors, which are supposed to prevent the cancers from growing. And one of the key characteristics of cancer is the ability to spread to distant organs or metastasis. This process is influenced by both genetic predispositions and environmental or lifestyle factors such as smoking, alcohol, diet, radiation exposure, and certain viruses. In fact, a recent advisory from the US Surgeon General emphasized that alcohol consumption increases cancer risk, including breast cancer. So why should we talk about breast cancer? Why focus on breast cancer? Because it's now the most commonly diagnosed cancer worldwide, surpassing even lung cancer. And while the death rates for breast cancer have been declining, it still remains the leading cause of cancer death in women. Let me share some sobering statistics with you. So 1 in 8 women will be diagnosed with breast cancer during their lifetime. And here in the US, younger African American women face higher risk and mortality compared to white women. And importantly, risk and outcomes do vary by ethnicity, and this is in part due to differences in tumor biology. So, understanding how we classify breast cancer is key to improving treatment and survival. So let's look at how breast cancer is classified. The classification starts with three categories, really. There's the classification into the type and subtype, which is based on cell surface receptor expression. And it's also classified based on stage. So the stage indicates how far the cancer has spread in the body. So stage 0, for example, would be precancerous cells, while stage 1 to 3 indicate cancer that is confined to the breast. And stage 4 indicates advanced cancer that has spread to other parts of the body. Cancer is also classified based on the grade, which describes how abnormal the cells look under the microscope. So a low grade, like grade 1 cancers, would look more like normal cells, while the higher grades, like grade 3 or grade 4, look very abnormal, and they also tend to grow fast and are more aggressive. So what truly defines a breast cancer cell is its receptor status. So here we're talking about the estrogen receptor and the progesterone receptor and HER2, which stands for the human epidermal growth factor receptor 2. If these receptors are present on the surface of the cell, it can be targeted by specific treatments. So tumors with the estrogen receptor or progesterone receptor are known as hormone receptor positive or HR positive, while tumors that have high levels of HER2 protein expression are known as HER2 positive tumors. And we'll be talking about this, as we go along in this talk. So these classifications guide treatment decisions and influence prognosis. So, and as I'll show you later, next generation sequencing is transforming how we define these subtypes altogether. So this slide shows what we understand about the genetics of breast cancer. While most breast cancers occur sporadically, spontaneously, without any known cause, there is about 10 to 15% that is due to inherited mutations or what we call germline mutations. You've probably heard of BRCA1 and BRCA2. These are the most well-known mutations that can cause breast cancer. But there are other germline mutations too, like PALB2, BRIP1, PTEN. Discoveries of these genes have led to targeted therapies. For instance, there's a treatment called PAP inhibitors, which are used in patients with mutations in DNA repair genes like BRIP1. So researchers are also comparing the genetics of the primary tumors where the cancer started and comparing that with their metastatic outgrowth to uncover how resistance to treatment evolves over time. And if you're interested, I've included a link in this slide that to an article that summarizes some of those key genes that are involved in metastatic breast cancer. In terms of treatments that are currently available, here they're listed. We have surgery is a treatment modality, and you have radiotherapy, chemotherapy, targeted therapy, and immunotherapy. And if the cancer is hormone receptor positive, as I mentioned earlier, then hormone therapy is essential. But even with all these treatments, there's still some unmet needs that remain. And chief of them is treatment resistance. And this is where the tumors suddenly stop responding to treatment over time. And there's also a need to identify the right order to deliver these treatments, these multiple treatments that we have available. And in terms of biomarkers, we need better tests to predict who will benefit from what treatment. And there's also the unmet needs of the side effects because many of these cancer treatments are hard to tolerate. So this is where genomic medicine has enormous potential to guide therapy, to minimize side effects, and to improve outcomes. All right, here's a quick timeline of how we've advanced personalized therapy in hormone receptor positive breast cancer with the help of genomic sequencing. And so this slide shows our long road to where we are today and how the therapeutic landscape has changed over the past few decades. Indeed, personalized therapy for hormone receptor positive breast cancer has come a long way, beginning back in 1896 when oophorectomy was used to reduce hormone levels. Oophorectomy, by the way, is a surgical removal of the ovaries. And if you fast forward to the 1970s, tamoxifen became the first selective estrogen receptor modulator that was developed to block the estrogen receptors. And at that same time, these chemotherapeutic agents became widely used in the clinic. And by the 1990s, aromatase inhibitors, another class of drugs for treating breast cancer, entered the clinic. And in 2002, fulvestrant became the first selective estrogen receptor degrader that was approved for use. And beginning in 2015, we've seen the approval of three selective CDK4/6 inhibitors, and they're palbociclib, ribociclib, and abemaciclib. These CDK4/6 inhibitors are they've become the established first-line standard of care when they're used in combination with endocrine therapy. And in recent years, we've seen new targeted agents that have been approved to expand treatment options for patients with breast cancer. And so these include targeted agents like inavolisib for patients that are harboring mutations in the PIK3CA gene, and elacestrant for patients who have ESR1 mutations, and capivasertib for patients who have AKT pathway alterations. So each of these steps has brought us closer to treatments that are tailored to the unique genetic makeup of each patient's tumor. Let's talk about the breast cancer signaling pathways, because if we were to fight breast cancer, we have to understand the cell's internal wiring. That is, how do the cells signal to each other and determine when to grow, when to spread, and when to evade cell death. And so the key signaling pathways that have been implicated in breast cancer are listed here. We have estrogen receptor signaling, driving some of the tumors and the excessive cell division. You have HER2 signaling, CDK pathway, PI3K, AKT, mTOR pathway, and the RTK RAS MAP kinase pathway. And in green, you're seeing the approved drugs that have been developed to target these pathways and block these pathways. While the pathways that are listed in red are still under research, we have not yet been able to develop treatments to intervene and disrupt those pathways, at least not for now. So let's focus on the CDK pathway, and I'll briefly describe the CDK signaling pathway using this simplified overview of the cell cycle and relevant upstream signaling components. A lot of you who have taken biochemistry would remember your cell cycle. And here we know that the most tightly regulated transition point in the cell cycle is your G1 to S transition point. Because that is where DNA gets transcribed into RNA. And here you see how extracellular mitogenic signals like estrogen or estradiol result in upregulation of these D-type cyclins. And these cyclins then form complexes with the cyclin-dependent kinases 4 and 6. And these active cyclin CDK complexes then phosphorylate the retinoblastoma protein, RB. And the retinoblastoma protein, RB, is a tumor suppressor. And when it becomes inactive, it releases the E2F transcription factor, which drives the transcription and synthesis of multiple elements that promote the G1 to S transition and further cell cycle progression. And so this is what results in cell proliferation. And in a nutshell, this would be one mechanism that's responsible for cancer. And as I mentioned, resistance to CDK4/6 inhibitors, which is a type of treatment for breast cancer, is a major challenge. But thanks to genomic tools, we're beginning to understand this more clearly. And we can see that resistance arises through cell-related mechanisms like the loss of FAT1, which upregulates CDK6, or the loss of RB, which bypasses the CDK control that we just talked about. We can have an overactive cyclin E and CDK2, and also the elevated aurora kinase A protein. And in terms of oncogenic signaling pathways, we see the RAS MAP kinase pathway and the AKT pathway activation as key contributors to treatment resistance. And on this slide, highlighted in yellow, are opportunities for us to intervene with currently available therapies or therapies that are still under development. All right, so let's now explore how genomics has driven innovation in oncology across drug development and treatment. And I'll walk you through some concrete examples that highlight this transformation. So since Mendel discovered the genes, one of the fundamental questions that we've always been asking is, how do we identify all the genes involved in human disease? And the answer really lies in studying the genome, which is the complete catalog of the human genes. So sequencing the entire genome has moved us from understanding isolated genes to building a full encyclopedia of genetic information. So most diseases, including cancers like we've just discussed, they are caused by disruptions in gene function. For example, here you see a point mutation in the beta globin gene on chromosome 11 disrupts protein function, and that results in beta thalassemia. So this journey from mutation to dysfunction to disease phenotype is what forms the basis of modern molecular medicine. And it's really changing how we practice medicine and how treatments are given to patients. So by way of also introducing sequencing, so sequencing determines the exact order of nucleotides in a DNA or RNA. We've talked about at this workshop, some of the earlier methods of conducting sequencing, like the Sanger sequencing, which was very slow and quite a manual process. Today, we have automated technologies like next generation sequencing, which enables fast, accurate sequencing of entire genomes. So, in terms of sequencing the DNA, the whole genome sequencing maps the complete DNA, including both the coding and the non-coding regions. And in terms of sequencing RNA, RNA sequencing, or RNA-seq for short, analyzes the RNA to identify gene expression patterns. And this can include whole transcriptome, targeted RNA-seq, and single-cell RNA-seq. So for those who are still new to these concepts, one way to think about it is that, your next generation sequencing is like your car. And whole genome sequencing is the driver that's sort of driving that car to explore the landscape of DNA that we're looking at and trying to understand. And likewise, for RNA-seq, it's a tool, and its applications like single-cell RNA-seq and are the destinations that this can reach. All right, so this slide shows some of the key milestones in genomics and highlighting a brief timeline here to show us the breakthroughs that have occurred in the past 75 years that has led us to where we are today. And back in the 50s with the discovery by Watson and Crick of the structure of the DNA double helix, fast forward to the 70s when Genentech was founded and recombinant DNA technology was born. And then by the 80s, we were still quite limited in what we could do in terms of sequencing. We could only sequence viruses. And by 1986, the first semi-automated DNA sequencer was developed. And by the 1990s, between the 1990s and 2000s, we saw the launch and completion of the Human Genome Project. And from 2000s to now, we have seen next generation sequencing emerge and the technology mature. So it's still a very new technology in the grand scheme of things, which really gives us in the African continent a huge opportunity to get in now and really own this technology. So these breakthroughs is what has laid the groundwork for today's genomics revolution. I would like to speak briefly about some of the genomic testing strategies because in cancer care there are various genomic testing tiers that exist. One way to think about it is that you would have your basic and your intermediate and your advanced tiers of testing. Your basic would be like your SNP genotyping, and intermediate would involve hotspot panels and DNA microarrays, while the advanced level of testing tier would involve the comprehensive genomic and transcriptome profiling. So these are some of the these tests can detect these mutations, which are all listed here. So you have your point mutations, indels, structural variants, composite biomarkers, even like the tumor mutation burden and the microsatellite instability. All right, so what sequencing has really helped us understand is that cancer is a disease of the genome and the epigenome. And there are many levels, several levels that we can look at this. We can look at it from the DNA level, and we can see from looking at the indels, copy number variations, and so on. And we can also see this by looking at the chromosome level, where we see amplifications, we can see rearrangements, and we see gene fusions occurring all through sequencing. If we look at the RNA level, we can also see fusion transcripts, for example, ALK EML4 shown here. And then if you look at the epigenome level, we can see from the methylation patterns and super enhancers, again, all genomic insights, highlighting that there's a lot of heterogeneity and complexity when you're talking about tumors, cancerous tumors. And this really underscores why personalized cancer care is so essential. So in brief, this is just showing us how the various technologies that are used in obtaining the tissue sample and sequencing it, and then using it to detect or to sequence those genes as we discussed. Typically, you start by taking a tissue biopsy, and then you may need to run multiple tests in order to be able to make a diagnosis. But the risk here is that if you have to run through so many tests in order to make that diagnosis, you may actually exhaust the sample and you might run out of the small sample that you were able to collect and preserve, whether it was an FFPE sample or frozen or it was a fresh sample. So we need a more efficient method of being able to obtain these samples and run these analyses. And this is where liquid biopsy comes in, using circulating tumor DNA. And this offers a non-invasive alternative method that enables frequent and real-time monitoring of the evolution of the patient's tumor without necessarily relying on a tumor sample. So let's talk about it a little bit more. And here I'm just highlighting the clinical utility of ctDNA. And the key things to highlight here is that it does allow us to monitor tumor progression and treatment response. And we're able to detect when treatment resistance is beginning to emerge. And that can help guide therapy decisions, especially when those tissue samples are not available. So ctDNA is becoming quite a powerful tool for precision oncology. All right, I just wanted to put this slide here and have us revisit it to remind ourselves of the traditional drug development framework that we talked about at the beginning of this talk, because genomics is really redefining how drug discovery is done. And just to put things in context for you, drug discovery and development is quite a risky business. And it's very expensive. It would cost about $2 to $3 billion on average to develop a single drug, and the failure rates are very high, something in the range of 85%. And part of the reason for this is because of poor target validation and our reliance on indirect biomarkers. And this is where genomics again offers new possibilities, because we can target gene regulation in not just the mutated proteins, not just signaling pathway in the cell. And by targeting gene regulation, we can expand the range of druggable targets that are available. And this is where epigenomics comes in, which involves those chemical modifications to the DNA and histones that allows us to modulate the gene expression directly. And one concrete example for you to take away is in acute myeloid leukemia, AML, where about 30% of the tumors have super enhancers around the RARA gene. And that's a vulnerability that we can now target with drugs. So, despite these advances, there's still many challenges that remain. One of the key challenges still remains that the tumors will continue to evolve under therapeutic pressure, and you still need to obtain these sequential biopsies, and you need to perform some sort of integrative omics. It's critically important in order to really understand what is going on with the patient. But this is very, very costly. So we need to solve the cost issue and make this scalable in a way that it is affordable and also accessible to everyone. We need a lot of genetic counselors to help make sense of the data that we're acquiring, and we need a lot of bioinformaticians. And we also need to find a way to integrate the data with electronic health records. And this is a very complicated task to do. And it's still a massive undertaking to translate all these mountains of data that we've been getting into clinical decisions. But in order to overcome these barriers, we do need a lot of investments in training and policy changes as well. And Africa has its own unique set of challenges in terms of leveraging genomics. One of the biggest issues is the limited access we have to NGS testing, and a shortage of bioinformatics talent, in the continent. And our efforts is also greatly undermined by the weak national science policies that we have in many of the African countries. And we also see huge underrepresentation in global clinical trials. So these are some of the challenges we need to solve for, and the opportunities that exist are also quite enormous. And I'm really happy and excited to see initiatives like the African BioGenome Project and H3 Africa building this foundation. Um, but we still need to do a lot more. We need to build population-specific variant databases. Africa is such a huge continent with, huge number of people, and that is an asset in itself. And we need to launch innovation hubs and local biotech startups. And we need to find a way to forge international partnerships, and lead in terms of pharmacogenomics, which I didn't really get the chance to get into in today's talk, but I did include a slide here that talks about pharmacogenomics for those that would want to take a look at that later on. We also need to lead in the areas of rare diseases and cancer biology, especially given Africa's unmatched genetic diversity. So this is my last slide and my call to action. And to really set the context for everyone, Africa will likely be home to 40% of humanity by the end of this century. Think about that for a moment. This continent is going to host 40% of all humanity. So we do need to find a way to invest in sustainable genomics, sustainable development, because if we are able to do that, Africa's future can be transformative and our power will be enormous. Um, and if we're not able to achieve this, it's hard to, it's hard to it's hard to even think about or even talk about the cataclysmic levels of economic and environmental breakdown that we would face. So it's imperative that we find a way to succeed in this endeavor. And bringing it back to healthcare, if we have to truly decode cancer across Africa and across the world, we must integrate genomics into routine clinical care. Again, precision oncology is no longer an experimental approach. It is a global standard, and we need to democratize that access right now. We need to do that by training the next generation of healthcare professionals in the continent. We need to build these genomic data systems that we've been talking about. We need to support African-led research. And if the policymakers and if the leadership and the government is failing, I would hope that private investors, private citizens, some of our billionaires can step in and show a good example, and then hopefully the leaders, our policymakers, will come around. Africa has a lot of talent, diversity, and the drive to lead, but we really must act now. And so to the students and the scientists in the room, I hope that you continue to pursue genomics, collaborate, publish, and lead. And to the policymakers and the funders who are listening to this, I hope you continue to invest and invest even more in Africa's genomic future. The future of cancer treatment, as I've tried to show in this talk, is written in the DNA. Let's ensure that Africa is part of that future so that together we can decode cancer and ensure that no one is left behind. Thank you very much. And this deck does contain a link to some useful resources. And I also thank some of the organizations that have supported my journey to this point. And I hope we can connect. Thank you.
Thank you so much, Dr. Obong Peters. I'm glad you didn't forget where you came from University of Calabar, NDCC, and then all the way. So it's really interesting to see the progression. And so we're going to kind of take some questions and then open up the floor for other comment contribution. So if anyone does have any comments, please do use the icon, the Zoom icon, to kind of indicate you want to ask a question or put the question in the chat box. So I'm going to go through. Does anyone, first of all, have any question orally to ask orally? Please do raise your hands. Any question oral? Otherwise, I'm going to go through the chat box to see if you do have any question. So someone is saying, um, Kemlavi, are you on the call? So I can ask you to Kemlavi. Is Kemlavi? No, it's not. Komlavi actually. Komlavi. Yeah. So I'm gonna ask you to unmute. That's Zenyo. Could you please unmute? Komlavi. I hope I'm pronouncing it right. All right. Please ask your question.
Okay, good morning.
Good morning. Go ahead, please.
I'm not really good in English, but I will try.
Okay. I don't know if we have what do you speak French?
French, yes.
Yes. So do we have any French speaker that can translate for us? Otherwise, we can. Any French speaker, please indicate via the message. Otherwise, please do ask your question in English, and then we'll try to understand it.
Okay, according to the lecturer of Doctor, uh, breast cancer comes from a modification of the structure of the cells. In this case, is the treatment specific to each case, or what is the principle of the choices of the medicine in front of each case, in each case of breast cancer?
Okay, so let me try to read it here. Dr. Obong, he said the breast cancer comes from modification of breast cells, which can be different for any woman. Is the treatment specific to each case? Or
Yes,
epidemiologically, every breast cancer must have certain types of modified cells, which are targeted by certain class of medicines. Briefly,
yes,
what is the principles of the choice of the medicine in the front of in each case? Dr.
yes.
Obong, I hope
Yes.
you, yeah.
Yes, I have a good sense of where you're coming from. So I would take it a step further and say it's actually the DNA in the cells that is different from one patient to the next. And so back in the day, we would use a one size fits all treatment approach and deliver, for example, chemotherapy. And these chemotherapies would attack the cells really, and they would have a lot of terrible side effects. Some patients would do poorly on them. Some patients, might fare better than others. But that was really the way we were treating it. Fast forward to today, and hopefully you can appreciate from the presentation that we had today that by applying genomic sequencing, we're able to detect the specific mutations that each of these patients carry. And there's some that are more common, like, for example, the PIK3CA mutation. It's present in about 30% of patients with breast cancer, with the HR positive, HER2 negative breast cancer. And so we are now able to provide them with targeted treatments. For example, we can prescribe inavolisib, which is quite selective for that type of mutation. And you combine that with the CDK4/6 inhibitor plus an endocrine therapy, and that provides better outcome for those patients. And it's a lot better than how we were treating breast cancer back in the 70s, for example, where everyone would get a selective estrogen receptor modulator, or everyone would get chemotherapy. In the future, what I'm hoping to see is that we would be using more and more these genomic sequences to, through modalities like ctDNA or even next generation sequencing to determine through pharmacogenomics what types of treatments we should offer to the patients so that we would have a more personalized treatment for each patient.
Okay, thank you, Dr. Ubong. You have one participant who would like to ask a question. Eric Wengi, could you please ask a question? You can put on your video as well, unmute as well. Can you unmute? You have, yes.
Yes, thank you so much for the insightful presentation. My question is, from my understanding, the breast cancer, there are different types of breast cancer. And from those different types of breast cancer, when you're doing treatment for those for breast cancer, you have to do diagnosis. And when you're doing diagnosis, do you look the biomarkers that you are targeting? Is it the same across those different cancers, or it's you look at different biomarkers for those types of breast cancers? Thank you.
Yeah, so the question is asking whether we would, use the same type of approaches to diagnose breast cancer. So I think if once you get these slides, you'd be able to see, I think at the very beginning of the talk, I sort of showed out how cancer would be classified. So that is, one of the ways that you would start by essentially just looking under the microscope and seeing how abnormal the cells looks like compared to a normal healthy cell. And you can also look at the receptor expression. And so there are modalities like FISH. And some of them are not that as sensitive as then using something like next generation sequencing, where at that point you're looking at the genetic makeup of that patient's tumor. And so you're looking at the sequences of nucleotides and determining whether the patient has a specific type of mutation that would then put them in a different molecular category where they would respond to better to a different type of treatment than, say, someone who did not have that specific type of mutation. So yeah, so to answer your question, it's a more complicated process now, I would say. If you wanted to really phenotype the genetic and molecular diversity of a tumor, there's a lot more involved in that than just looking under the microscope and determining whether a cancer cell bears the cell surface bears certain receptor expressions or not.
Thank you, Dr. Ubong. It looks like you have a lot of interaction. So I hope you have the time for us today. Josephine Don Bosco has a question. Josephine, could you please speak? You can unmute, and you can also turn on your video if you want. Josephine Don Bosco, could you please unmute? Yes.
Yeah. Could you hear me?
Yes, please carry on.
Yeah, good morning, sir.
Yes.
Good morning.
Okay, good. My name is Jospin Awoliati. I'm from Benin. And my question is directly to you. I would like to know what is the role, or when you talk about next generation sequencing, what does it mean? And apart from that, how can we use that directly based on, use it on plants in order to find at least a medicine or develop a kind of medicine using plants in order to treat cancer or, any type of cancer?
Yeah, so when we talk about next generation sequencing, we are talking about a way of looking at the precise order of the DNA. So the DNA actually has both 4 different types of nucleotides. So you could have adenine, thymine, cytosine, and guanine, and we know that adenine binds to thymine, cytosine binds to guanine. So when you are looking at the DNA molecule, you're basically trying to figure out what order those sequences are. And so that's what next generation sequencing is doing for you. Let me see if I can find a slide where that shows this in a much better way. And so when you have changes in the where those sequences are supposed to be arranged, that is what could lead to a disease. So for example, you see here that in this particular chromosome 11, there is a point mutation where instead of having a T, this has changed to a C here. And this is what has caused a patient to have a specific disease. So that is what the sequencing is doing for you. It's helping us to understand the exact order in which these nucleotides are expressed.
Thank you, Dr. Obon-Peters. Jasmine, I hope that clarifies your question. So we're going to move to the next speaker, the next participant who wants to ask a question. Jolie Thomas, could you please ask your question? You can unmute as well. You can unmute Jolie Thomas. Yes, carry on.
Okay, thank you very much, sir. My name is Jolie Thomas. I'm from the College of Medicine, the University of Ibadan. So my question, sir, is see, for the fact that Africa is genetically diverse, like we are the most diverse race, but still, when it comes to research and access to funds, Africa is still way behind. So my question now is, how can cancer research be translated into affordable and accessible diagnostic or treatment strategy in low resource setting, especially in Africa, where the burden of late stage diagnosis is very high?
Yeah, so what I think the best solution in a situation like that would be to apply the economies of scale. When you have such a huge number of people residing in one continent, such as what you have in Africa, or even this projection that 40% of all humanity will be residing in Africa by the end of this century, that in itself, those numbers, that's our asset right there. Because a lot of the technologies that are being developed elsewhere, those technologies would need data. And it's really that data that, we're using AI to try to use that data to predict, to apply AI on that data to predict a lot of the things that are helping drive the revolution and the innovation in this type of work. So if we have the data, then it's up to us to figure out a way to negotiate a deal with the people who actually have the equipment and it doesn't mean that we are trying to just freeload on a lot of this, but there are ways that we can structure it in a way that also involves the country having some sort of payment that they make to the companies as well. And some of the healthcare budget would be set aside to pay for a lot of these tests. And we could also be building huge data repositories and registries that again would be a gold mine for people who actually want to mine that data and get insights from that data. And so that gives us additional leverage in our negotiation. So there are so many ways that we could try to position ourselves such that we still have some sort of bargaining chip that helps us lower the cost of this. But regardless of whatever way we use, it still has to involve at the national level some sound health policy that would involve setting aside a huge chunk of the budget, maybe the healthcare budget, towards buying a lot of these new medicines that are coming out, buying a lot of these technologies that are coming out, and training the next generation of healthcare professionals to be able to utilize those tests, and building the databases and figuring out a way to curate those databases in a way that it would be very attractive to people who want that data.
Thank you, Dr. Obong. So I'm going to collect every single question for you today. I hope you have the time. We have a question from Ifeoluwa. Ifeoluwa Oyeyemi, could you please unmute yourself? You can unmute yourself now. All right, carry on.
Okay, thank you so much for this insightful presentation. Thank you. I just want to ask, coming on now as a preclinical scientist, like most of the facilities, we are not as advanced in this area as in other parts of the world, and there aren't so much skills in terms of using organoids. But then I want to say, using in vitro cell line to study potential drugs, I mean drug molecules now, for that can target this therapeutic pathways that have been identified for cancer. Do you think it's a worthwhile exercise? Then I want to ask, most of the cell lines we use, we know that they don't encapsulate African unique genetics because they were not isolated from Africans. Do you think that working towards developing cell lines that are unique, isolated, and unique to Africans that can probably reflect African genetics, do you think it's a worthwhile exercise? Thank you.
Yeah, thank you very much for that question. Yeah, I think it is definitely the two questions you asked were really around whether it is worthwhile for Africa to be involved in this type of scientific research, preclinical, given that the setting there's very low resources available in this setting. My answer to that would be that we can't afford not to be involved in this type of research, and it doesn't mean that we have to do it at the level that the West is doing, because they may also have a lot of the resources and they can dedicate those resources to a lot more of those resources to preclinical scientific research. At the same time, there are things that we should be doing that we frankly can't just wait for the West to do for us. And the question around being able to perform research on diverse preclinical samples, cell lines that have been obtained from, or samples that have been obtained from people within the continent. This is where we're missing out if we're not able to perform that research in our country, in the African continent. So what I would like to see is a future where all of humanity is involved in scientific research, in medical research especially. It shouldn't just be research shouldn't just be done in the US and in Europe and China and elsewhere. It should be done everywhere. We all have to put our hands on deck to try to fight cancer, we have to all put our hands on deck and do whatever we can to try to find a solution to this scourge. And the way science also works, there's a lot of serendipity in science, where you might just discover something out of good fortune, out of luck. And so that one discovery may be enough to change the fortunes of the continent. So I would encourage everyone not to be discouraged by the fact that we are doing research in a low resource setting, but to continue to persevere, to do whatever we can, and to write and publish the knowledge. Whether it's a negative study, a positive study, you do your best to try to have that published. And one day that knowledge could help someone. It could help us unravel a mystery. It could help us solve a puzzle that has been plaguing us for a long time.
Thank you, Dr. Ubong. So while you're still here, Ifeoluwa, Oyeyemi, and Uduak, you guys will be presenting this morning. So please hang around still be on the Zoom, so that we have, after Dr. Ubong, we're going to go to one speaker, and then Ifeoluwa followed by Uduak. So Dr. Ubong, we have one more question before we go to the participants on site in Port Harcourt. Anita, do you want to yes, Anita. Oh, no. Anita, could you please raise your hand again? For some reason, I lowered it while trying to unmute you. Anita? Or did you lower it intentionally? Anita. Okay. Can you unmute now? Anita? Yes. Can you unmute?
Okay.
Yeah.
Can you hear me? Good
Yes,
morning,
please
everyone.
go ahead.
Good morning, sir. Okay. So, Dr. Okon, sir, in the case of the point mutation that you showed us in the slide, has any research been carried out to reverse such mutations? Yeah, has any research been carried out? Okay, just generally, has any research been carried out to reverse point mutations in a human genome?
So I can't say confidently whether it's been specifically done for point mutations, but there's a new class of therapies that are coming out. In fact, a few of them have already been approved by the FDA. They're called gene therapies. And that's been, using some sort of a vector to go into into the gene to try to correct some of those mutations. And so, yes, there are a number of gene therapies that are already approved out there. And we know that with CRISPR and so many other tools that are coming out that may even be coming out in the future, more and more of such therapies would even be more available in the future.
Thank you very much.
Yeah.
Thank you, Dr. Ubong. To the participants on site at the University of Port Harcourt, Control, do we have any question for Dr. Ubong Peters? Please. Yes, please.
Yes, so please, before I give the mic to people who want to ask questions.
Dr. Ubong, can you please stop sharing so that we can see them? Stop sharing the screen. Yes, thank you. Yes, please.
here.
We can't hear you. So control. Yeah, we can hear you now.
Dr. Komi Pokora is in Lome. He's our translator. He does all the language translations between English and French. So in case any person wants to ask a question in French, the person should ask freely because Komi is online to do the interpretation.
Okay.
Then I also want to announce that the project coordinator of HyPrep could not be here, but he has sent a representative, Dr. Obari, who has come to represent him. In due time, he will give his speech. But for now, please, I want to give room for the people to ask questions. Please, you are the first. Can you come and ask your question, please?
Good morning, Dr. Obong. Thank you for your presentation. Can you hear me?
Yes, please carry on.
Okay, my question is this. To a layman who is not so knowledgeable about the use of molecular markers or testing for specific drugs to administer to your patients, and the person walks into your clinic and wants to get treated for cancer, and you want to go through all this long process to be able to decide precisely which drug to administer to the patients. And knowing that cancer is very aggressive and can spread very widely and kills fast, how long does it take for you to go through all these processes for you to be able to decide which drug is specific or would work effectively for that particular patient? And also knowing that some drugs can be resistant. And so if a drug is resistant, do you also go through a second cycle again to be able to get through the DNA to know which drug will now work very well for that patient? That's my question. Thank you.
Yeah, that's a really good question. Thank you very much for asking that question. The first question you asked was, how do we find answers for the patient very quickly so that they can start their treatment? And, because oftentimes if we catch the disease early, like for example, if it's early breast cancer, the intent of treatment would be curative, and in fact, most of those patients can be cured if the cancer is caught early. So it is very important to catch it early. However, when you talk about trying to figure out what is going on, depending on the types of tests that's available to you, whether you're doing a single panel or microarray testing, you may have to rely on a small sample. You might take a slice of your sample and run it, and you may be able to get some answers from that sample. But if you don't, you might have to do it a couple of times before you can actually arrive at your answer. This is where next generation sequencing or doing a comprehensive genomic profiling is a lot better, because you would be able to get at that answer very quickly. So you do the whole genome sequencing all at once, and you can actually get those answers, and it would save you the time of having to run multiple panels to try to get at the particular issue that's wrong with the patient and what sort of treatment would work. So the only downside here is that it is expensive. It's a lot more expensive running this whole genome sequencing than just doing a few panels. And so that is where we need to try to strike that balance. And I think it's a huge problem for patients in Africa, because a lot of the patients who are dying needlessly there, some of them, if they were in places like here in the US, a lot of those patients would be saved, because they would have access to these types of tests. And so doctors would be able to detect it very quickly, and they would be able to provide the treatment for them. So this is a topic that really bothers me a lot because there's just so much more that we need to be doing. And it's not just the government that is to blame. I think even private individuals here are to be blamed as well, because if the government is failing us in, trying to make it, provide the resources for us to be able to access some of these tests or these treatments, then private individuals should be able to step in. There are a lot of wealthy Africans, billionaires in the continent, who could actually be leading the way and showing that it's possible to have, in a country like Nigeria, maybe you have 6 centers that have, very good laboratories that can run whole genome sequencing, the RNA-seq, everything, whatever test you need, that they have the capability to be able to run that. And then the treatments as well. When the pharmaceuticals develop these treatments, they're not going to sell it for free to us. It takes about $2 to $3 billion to generate each of these, or to develop each of these treatments. So our national health insurance scheme has to figure out a way to pay for this, so that the common man can afford this treatment. So it's a long-winded way of trying to answer this question, but I think there's a lot that we can do. It all comes down to cost and access, and this is where we need resources, but it's not coming from the government. And if it's not coming from the government, I think private individuals need to step in.
Thank you, Dr. Ubong. I will let you go, but 2 more questions. Well, I'm going to merge the questions into one. So you actually just answered one of the questions. Peter Holland Maman is asking if Africa can afford personalized treatment, but I think you answered that in your last comment. So I'm just going to go to Favor. So this is now the online chat going on here. I'm going to go to Favor Edie. She's asking, considering the challenges in Africa, what's your advice on accessing resources? Again, this is like career. She says she's also looking forward to a postdoc, and what are the opportunities? So I think beyond this question, I would like you to kind of finalize, because this is my last question to you. How do you you need to slide your responses into career, and given that we have lots of early careers, mid careers, even professors on this meeting, and lots of them are on our site and on the Zoom call. So you can respond to her question and then see how you can slide it. And of course, you've done. I've seen you introduce yourself and where you started, the days of little beginnings. So just let us spend the next 5-10 minutes, please.
Yeah, so there's a lot to unpack in that question, because here we're talking about next generation sequencing, and I highlighted some of the key challenges that we're facing in trying to make this technology a reality, or even harness the full potential and ensure that Africa is leading when it comes to next generation sequencing. To be able to do that, we need a very strong workforce. And that strong workforce is in our youth. We need a lot of genetic counselors. We need a lot of bioinformaticians. We need a lot of scientists. We need we need people who understand computer science and can crunch the data and help make all of this a reality. And so to be able to get that knowledge, it's great that there's a lot of interest and there's a lot of engagement based on the number of people attending this program and even the kinds of questions that are coming in. So yes, whatever you can do to get into a program in biotechnology, in genomics, genetics, and just learn, because at the end of the day, we do need to educate the public. We do need to educate our policymakers, because it could be that they don't really understand why this is so important. And if they do understand why it is important, they would be able to create the resources necessary for the youth to take advantage of. So my own story, for example, I think is very poignant here, because it was the Akwa Ibom State government that provided a scholarship through former Governor Victor Tar that allowed me to go to Canada to study for my master's and my PhD. But even then, there were a lot of struggles within the government, because as soon as Governor Tar left and Governor Babio came in, he had a different direction he wanted to take the state. Not to criticize any of his policies, but he did not continue that program that Governor Tar started. And so there is no scientific leaders. There are no other young people who have joined me out here or done this type of research, just like I did. And so it's just me doing this now. Whereas we had a government sending out, even if it's 10 students, even if it's 20 students, to go overseas and study, and they can one day turn around and inspire others. This is very important. I know at the end, this is doing providing these types of scholarships. I don't know if they're still providing these scholarships at very vast numbers that would really make a difference. But you do need this type of training to be made available to the students that are in Africa. And we shouldn't necessarily worry about the fact that these students, some of them could go get training and then not return. That's not the case. Even for the program that I was part of with the Akwa Ibom State government, I would say that close to 40% of us went back and contributed. And they're working at the Akwa Ibom State University as we speak. And so what we really need to do is provide these opportunities for young people to go out, get the training, and some of them might find their own career paths, and most times those paths could lead them back home again, could make them inspire others. And even though I'm out here, I still have very deep affection and connection to my home, and I would want to give back at some point. And so the fact that I'm out here doesn't mean that I don't have plans to one day come back and give back to my community for what they've done. So I hope that answers your question, but in my closing remarks, it would be that regardless of how the situation is, the students should continue to be motivated, take advantage of whatever opportunities you find, work extremely hard. You do need to work 2 or 3 times or even 5 times harder than everyone else, because that's just what it takes to be able to set yourself apart when you come from the kind of environment that we come from.
Thank you, Dr. Ubong Peters. I have just been told that there's one more question on site, and I wanted to let you go so that we can move to the next speaker, and I can see people raising their hands. So we're going to do 2 things. We're going to ask the one question on site, and then please, Dr. Ubong, just stay behind and respond to any other questions on the chat box. Okay. And some participants also want your slide. Could you please share it with me, and then we'll share it with all the participants if you are happy to, because I understand sometimes, you just let me know. If you want to share it, just send it to me, then I will pass it on to them. Otherwise, you are not obliged to share it.
Oh, absolutely. This is all for public release. So yes. I'm happy to share those slides.
Okay, please do share it with me, and then we'll pass it on to them through the mailing list. We have about, for this conference, 1,500 registered, and some people join at particular sessions, and sometimes because of the bandwidth and stuff like this. All right, control the local participants on site. One question, please, so we can free up Dr. Ubong Peters. We can't hear you, please. Okay, go ahead.
Thank you very much for sharing wonderful knowledge on the scientific methods.
Could you please speak up? We can't hear you.
My question is on DNA sequencing. And DNA sequencing is about knowing the precise nucleotides order in DNA molecule. But in a situation whereby we have a mutation, for example, in sickle cell anemia, where we understand that the base pairing has the 9 pairs with thymine, but the so that's adenine pairs with thymine. Sometimes during mutation, the order changes. So if you want to do a DNA repair, how do we know the precise, the normal, so that we will now use the abnormal, we correct the abnormal from the normal DNA sequence of that patient? You understand?
Yes.
So the question is, we have to know the normal DNA sequence of the patient. When there's some mutation, when we do the DNA application, we see the abnormal. How do we now get the abnormal? So we have a wide range of DNA sequences. So how do we differentiate them? So this one is normal and this one is abnormal.
Yeah. So it's a very typical science question. And I think once you get my slides, you're going to see that there is on towards the end of the slide deck where I've included some useful resources there. There's some databases like CleanVar that contain all those databases that show what the what would be normal and what would be abnormal. But in a nutshell, there is what they call a reference library against which the sequences are being compared. So these reference libraries would be generated from huge sampling of the population to know, for example, what the vast majority of people would have at that, say that particular point, whether it should be ATC. So if you've measured 100,000 people and 99% of people have ATC, then it's easy to make a call when you see the 1% that have a different sequence that like TCA or something like that. So it's a scientific problem that you can solve very easily with math.
Thank you, Dr. Ubong. Will please, as I mentioned, do hang around and answer any question on the chat box. And sorry, Malaki, we have to really move ahead. Please do put your question in the chat box, and Dr. Peter Obong will attend to it. All right, thank you so much, Dr. Obong. We'll move to the next presenter.
Just 1 minute to just take one brief question from here.
Okay.
Yeah. Okay. Okay, thank you very much. My question goes this way. We have been talking about drug development, and there's some common things we observe in Africa, especially in Nigeria. You have a situation where people talk about some foods, herbs that are anti-cancer, and you see the multiplying in the social media. People missing this and missing that. Actually, I did some work in coconut water to actually find out some of the anti-cancer effects. So the question I want to ask, this drug development, which you have presented, does it follow the same pattern if you want to use some of these natural foods to find out whether they have anti-cancer from there to develop a kind of drugs for cancer? Do they follow the same pattern, or it's a different route when we are doing the economic sequence? Thank you, sir.
Yeah, thank you very much. That's also a very good question. And the short answer would be no. In some cases, yeah, maybe you could rely on some plants to be able to figure out what the active chemical properties of that specific plant might be that might confer the properties that it has. So for example, if there's a specific plant that has antimicrobial effects, you might do some sort of phytochemical analysis to see and to isolate the specific chemical or to know what chemical structure for that molecule looks like. But it's usually not the natural product that the pharmaceutical would end up packaging as a drug in the end. We would have, chemists, we have biochemists, we have, all of these folks who can go into the lab and they can take that natural plant that let's say, for example, there's a specific leaf that we know has antimicrobial properties. It's not the leaf itself. There is a specific molecule in that leaf. And so once you do the phytochemical analysis and you really drill down, they would finally isolate that one specific chemical, that one specific molecule driving those effects. And then someone can actually synthesize it in the lab using the basic structure of any chemical. It could be nitrogen, it could contain some carbon rings and some hydrogen. They'd have some really fancy models that they can use in synthesizing that molecule. And then they can keep tweaking it as well. They might decide, oh, what happens if we add a methyl group here? What happens if we add a hydroxyl group here? Does it make it more potent? Does it make it more selective? Does it make it more efficacious? So there's so many different things a chemist could do around, play around with the molecule in order to actually know what the effects would be. And then once they have zeroed in on a lead candidate, that is what gets that is what moves in the pipeline to the next stage of development, where you start testing on animals and cells. And if that molecule still proves to be effective in killing the cells in the specific way that we want it, and also effective in animals, and it's not affecting the organs of the animals, then we might decide to try that molecule in clinical trials. So it's a completely different process, and I would caution against relying on, some of those what I would say anecdotal evidence of people saying, "Oh yeah, if you consume grapefruit, it will cure cancer." It would do. We've seen that happen back, as I started in my talk, back in the pre-industrial era, how people were marketing all these products like Wizard's oil and saying that it cures everything. It's just not true. It's just not true. You have to actually do the science to see whether it works. And human beings are also very complicated, that even when you're doing the trials, and you sometimes you have to design these trials in a way that you are dividing the groups of people into two. On one half of the people, you give them the actual drug that you've developed, and the other half, you give them a sugar pill. And yet, sometimes we see in the people that receive the sugar pill, they still receive some sort of clinical benefit. It's like the sugar pill did something for them. It's called the placebo effect. So human beings are very complicated in that nature, that even when they believe that something they've taken is effective, sometimes it works for them. It's a placebo effect, but it doesn't necessarily mean that this is a scientific effect, or this is an effect that is reproducible in the vast majority of people. So I would really caution against unproven claims like that do not have any scientific backing.
Thank you, Dr. Ubon-Peters. We would like to really move to the next speaker. So please hang around and then comment on the chat box. Thank you so much. Could you please put a hand up, applause for Dr. Ubon-Peters, and those on Zoom, you can use the Zoom, icon for clapping. All right. The next speaker would be Olario Waju. hang on. I'm just gonna. Yeah, Olarewaju Alofowobi from Unkaba Biotech. Larry, you can now share your screen, please.